Hello, everyone, and welcome to today's presentation, part of the Agilent Cell Analysis Global Conference. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Ask a Question box located on the far left of your screen. I now present today's speakers: Kristin Bircsak, Ph.D., Principal Scientist, MIMETAS, and Kristen Olson, Ph.D., Imaging Specialist, Field Application Scientist, BioTek Instruments, now a part of Agilent. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Drs. Bircsak and Olson, you may now begin your presentation.
The title of our presentation is MIMETAS and BioTek Instruments, a toolbox to dissect the vascular impact of COVID-19. This webinar is a collaborative presentation by Dr. Kristin Bircsak, a principal scientist for MIMETAS, and Dr. Kristen Olson, an imaging specialist and field application scientist for BioTek Instruments, now a part of Agilent. So first, let's discuss a bit more about the virus and the vessel. COVID-19 is an emerging zoonotic disease that has resulted in the current pandemic and ongoing global health crisis. The first cases of the disease originated in Wuhan City in the Hubei province of China in December 2019. And owing to its highly infectious nature, the disease has spread rapidly around the globe. At the completion of this presentation on July 12, 2020, the World Health Organization reported over 12 million infections worldwide. Of these 12 million infections, over 3.16 million were in the United States, accounting for 25% of the global infections at that time. Of those 12 million global infections, over 500,000 resulted in death, with over 130,000 fatalities occurring in the United States, accounting for 24% of the global COVID-19 related deaths. As of today, just over 3 months later, the number of global infections has grown from 12 million to over 38 million, with over 7.7 million confirmed cases in the United States. And globally, COVID-19 has been the cause of over 1 million deaths, with just over 213,000 deaths occurring in the US. Owing to the severe impact of this disease throughout the world and the high percentage of morbidity and mortality, it is of the utmost importance to understand the pathological mechanism of this disease. The causative agent of COVID-19 is SARS coronavirus 2 or SARS-CoV-2, a positive sense single-stranded RNA virus and a beta coronavirus in the family Coronaviridae. SARS-CoV-2 is one of 7 human pathogenic coronaviruses. Whereas 4 of these pathogenic coronaviruses typically cause mild cold-like symptoms, the remaining 3 pathogenic viruses, SARS-CoV, MERS-CoV, and now SARS-CoV-2, can cause severe disease, with SARS-CoV and MERS-CoV resulting in epidemics in 2003 and 2012, respectively. During infection, SARS-CoV-2 enters into host cells by both receptor-mediated entry and receptor-independent plasma membrane fusion. During receptor-mediated entry, the viral spike glycoprotein binds the angiotensin-converting enzyme 2, or ACE2 receptor, that is expressed on a wide range of host cells. While SARS-CoV also binds the ACE2 receptor to facilitate entry and cause disease, SARS-CoV-2 binds at a much higher affinity. This increased affinity may play a part in the increased infectivity and worldwide spread of this novel virus. Additionally, the expression of the ACE2 receptor on a wide variety of cell types in multiple organ systems may facilitate the systemic spread of the virus in moderate to severe disease. SARS-CoV-2 can be transmitted both directly and indirectly from an infected host to a non-infected individual. Unlike many respiratory viruses, SARS-CoV-2 can be transmitted from an infected host even if they are asymptomatic or pre-symptomatic, making it very difficult to discern if any individual is contagious. In direct transmission, an infected host transmits the virus to a non-infected individual directly. In the case of SARS-CoV-2, this is mainly mediated by respiratory transmission of large, short-range respiratory droplets. These virus-containing droplets are generated when an infected individual sneezes, coughs, laughs, sings, or even talks. If a non-infected person is within range of these infectious droplets, they can be exposed to the virus via inhalation, depositing infectious virus into the upper and lower respiratory tract, or even exposure via the mucosal membranes of the eyes, nose, or mouth. The risk of direct transmission of SARS-CoV-2 can be greatly reduced by following social distancing guidelines and properly wearing masks when in public. Indirect transmission of the virus occurs when the virus is transmitted from the infected host to a susceptible individual indirectly. For SARS-CoV-2, this can occur via suspended small particle long-range aerosols or contamination of inanimate objects termed fomites. While there has been controversy over the aerosol transmission of SARS-CoV-2, the World Health Organization and Centers for Disease Control have updated their guidance, stating that in indoor spaces with poor ventilation, small particle infectious aerosols may remain suspended and pose a greater transmission risk than previously thought. In the case of SARS-CoV-2, fomite or vehicle transmission could occur from infectious droplets contaminating surfaces, such as doorknobs, office equipment, or even the card reader at the cash register. If a susceptible individual touches that surface and then touches the mucosal membranes in their mouth, nose, or eyes, they could become infected with the virus. The easiest way to prevent this type of transmission is to avoid touching your face and making sure that you're frequently washing your hands with soap. While fomites may contribute to transmission, they are currently thought of as playing only a minor role in the spread of the virus. Previous data suggested that additional routes of SARS-CoV-2 indirect transmission may occur from fecal shedding of the virus. While the virus and viral RNA have been isolated from contaminated wastewater, this is currently not considered to be a likely source of indirect transmission. Now that we understand the origins of this disease and how the virus can be transmitted, we should discuss the clinical characteristics of COVID-19. There has been great difficulty in characterizing the clinical course of COVID-19 due to the spectrum of disease and severity that occurs in different individuals. Some infected individuals may be completely asymptomatic, making it very difficult to truly know how many people have been or are infected. In addition, many cases exhibit only mild symptoms, typical of other respiratory viruses, including a low-grade fever, cough, fatigue, sore throat, body aches, and headache. These symptoms make the disease clinically indistinct from other respiratory infections. During the early stages of the pandemic, this may have also led to a reduction in infected individuals reporting or being tested for infection. In some individuals, mild cases progress to more serious symptoms, most commonly shortness of breath or dyspnea. This shortness of breath can result in a moderately severe pneumonia and may require hospitalization. In severe cases, respiratory symptoms may progress to acute respiratory distress syndrome, or ARDS, or other severe disease complications, including but not limited to acute kidney injury, cardiac injury, septic shock, metabolic acidosis, coagulation dysfunction, liver dysfunction, and secondary infection. In a percentage of individuals, this disease is fatal. While a majority of fatalities are associated with age or underlying conditions, we do not yet fully understand the reason this disease causes death in some individuals while causing mild or no symptoms in others. The disease course and timeline can vary, but in many individuals who will go on to experience moderate to severe disease, the timeline is as follows. Incubation, during which an individual is asymptomatic or presymptomatic, can last from 2 to 14 days. During this time, the individual can transmit the infection to others before knowing they are infected. During the symptomatic phase, an individual may experience severe symptoms such as dyspnea in the first 2 to 5 days. Typically, these individuals will require hospitalization within 2 days from dyspnea onset, and if this progresses to ARDS, the individual may remain in the hospital for 10 days or more. By 21 days post-symptom onset, symptoms will begin to decline in many individuals, and they will experience convalescence of the disease by 4 weeks after onset. While the disease course is an example, many individuals can resolve sooner than this, and some individuals will go on to experience sequelae and post-disease symptoms long after convalescence has occurred. So what has made SARS-CoV-2 such a successful pathogen? The success of any pathogen is defined by the following characteristics. Infectivity, or the capacity of a pathogen for horizontal transmission from one individual to another. Transmission, or how the pathogen passes from an infected individual to a non-infected individual. And disease course, the resulting pathology of the disease, the spectrum of disease experienced by infected individuals, and the tissue or cell types that are infected. Infectivity can be mathematically defined by the number of individuals infected by one infected host in the current state of the population, a number referred to as the effective reproductive number. When this number is less than 1, the pathogen will not spread, and when this number is greater than 1, the pathogen will spread rapidly. The current effective reproductive number for SARS-CoV-2, both globally and in the United States, is an average of 1.3, suggesting the pathogen will still spread rapidly. Factors that have contributed to the infectivity of SARS-CoV-2 include the availability of naive hosts. At the beginning of the pandemic, there was no pre-existing immunity to the virus, and currently there are still many naive and susceptible hosts available. Additionally, the virus has the ability to evade the immune response on multiple levels, lending to the inability of the immune system to quickly control the infection and prevent further spread. While pathogens are transmitted via multiple mechanisms, SARS-CoV-2 is a respiratory pathogen. While we are still understanding the transmission route of the virus, it has now been established that it can be transmitted by both short-range, large respiratory droplets in close proximity and long-range, fine particle aerosols in enclosed spaces with poor ventilation. Additionally, the virus can be transmitted by asymptomatic or pre-symptomatic individuals, making it difficult to discern who may be infectious. Together, these factors require that we practice proper respiratory protection and distancing at all times, not just around individuals that are displaying symptoms. And finally, the disease course of COVID-19 contributes to the success of this virus. Individuals can experience a very long incubation period during which they may be shedding virus before knowing they are infected. This long incubation time may also delay the individual from seeking testing following exposure and make contact tracing more difficult. Individuals experience a spectrum of disease, making the clinical distinction of COVID-19 difficult without appropriate testing. And the tropism of the virus, or the types of cells and tissues that the virus can infect, is not limited to a single cell or tissue type. Due to the distribution of the ACE2 receptor on many cell types and tissues, the virus has the capacity to spread systemically and infect multiple organ systems. As mentioned, there are many severe complications of COVID-19. Some of these complications are a direct impact of the virus, while others are the result of an unchecked immune response resulting in immunopathology. While COVID-19 is considered by many to be a respiratory disease, many organ systems are impacted during infection. In most cases, the lungs are the primary site of infection and typically suffer the most damage from the virus and subsequent immune response. The virus typically will enter the body through inhalation in the upper respiratory tract or mucosal membranes of the nose and mouth, then travel deeper into the lungs into the lower respiratory tract. The virus impacts the airways and site of gas exchange, infecting bronchial epithelial cells and alveolar epithelial cells. During infection, the virus can cause cell death, leading to a loss of structure and damage to the delicate gas exchange surface area. Additionally, infection and damage recruits immune cell populations, causing the airway to fill with fluid, debris, and inflammatory cells. The impacts to the cardiovascular system are both direct and indirect, and will be the focus of the remainder of this introduction. Although typically considered a respiratory disease, recent data suggests that COVID-19 may actually be a vascular disease. The vascular pathology caused by SARS-CoV-2 is a product of both the direct and indirect impact of the virus, shown here as 5 different levels, with each level contributing to increased vasculopathy, into endothelial cell death and subsequent denuding of the endothelium. The loss in the endothelial cell barrier allows fluid to leak into the surrounding tissue as part of this immediate impact. The indirect impact of SARS-CoV-2 infection on the vasculature is demonstrated in levels 2 to 5. The second level of vasculopathy occurs during the massive inflammatory response in moderate to severe SARS-CoV-2 infection. The remaining levels occur in patients with progression to sequelae associated with increased morbidity and mortality. In the third level, the massive inflammatory response and coagulation response to damaged vessels and resulting tissue damage can lead to tissue ischemia and hypoxia. This decreased oxygen can trigger an increase in angiogenesis or the formation of new blood vessels in the affected tissue. In advanced cases of COVID-19, this process is dysregulated, leading to deformation of the vasculature and a loss of vascular hierarchy. As the disease progresses and vascular damage and leakage occur, the coagulation cascade is upregulated, causing the formation of microthrombi and thrombi in blood vessels and tissues, further amplifying ischemia, tissue death, and contributing to hypoxia, stroke, or pulmonary embolism. With the most severe pathology, we see the 5th level of vascular impact, where severe cases have marked angiopathy, where the vasculature itself is diseased, causing dysregulated circulation and severe hemodynamic changes that result from all of the aforementioned impacts of the disease. Now that we have discussed these multiple levels of vascular impact during SARS-CoV-2 infection, we'll provide a brief overview of normal vasculature and pulmonary circulation. The endothelium is comprised of a single layer of squamous endothelial cells that line the interior of all blood vessels and lymphatic vessels. These endothelial cells have 3 main functions in maintaining homeostasis. They serve as a barrier between circulating blood within the lumen of the vessel and the surrounding tissues. Through this function, endothelial cells communicate with neighboring cells to regulate blood flow and maintain hemostasis. The endothelial cells act in paracrine, autocrine, and endocrine ways to limit immune cell and platelet interactions, and additionally via endothelial cell tight junctions, as shown in the fluorescent microscopy image below. The endothelium functions as a gatekeeper, regulating leukocyte trafficking and the access of plasma proteins to the surrounding tissues. The maintenance and regulation of the pulmonary vasculature is of utmost importance, as the lungs, and more specifically the alveoli, are the site of all gas exchange in the body. The pulmonary circuit is responsible for the oxygenation of all blood in the systemic circulation by transporting blood between the heart and the lungs. Following systemic circulation, the deoxygenated blood returns to the heart and exits the right ventricle into the pulmonary arteries, flowing into both lungs. In the pulmonary circuit, there is a defined vascular hierarchy, with the branches of the pulmonary artery splitting and following alongside the bronchial tree toward the alveoli, where each alveolus is surrounded by a vast capillary network. The alveoli are terminal sacs of the individual bronchioles, providing a thin but vast surface area for the vital function of gas exchange. The alveolar wall is comprised of a single layer of simple squamous epithelial cells, the type 1 and type 2 pneumocytes. Type 2 pneumocytes are responsible for the production of surfactant, a fluid that maintains surface tension within the lung. When damaged or lost, the type 1 pneumocytes can differentiate into new type 2 pneumocytes, maintaining the production of surfactant and preventing alveolar collapse. Beneath this epithelial surface lies a basement membrane comprised of extracellular matrix, a thin interstitial space, and then the endothelial cell wall of the alveolar capillary. These layers together are termed the alveolar membrane and are the site of gas exchange within the body. As the alveolar wall is comprised of only a single cell layer, the alveoli are easily damaged. Loss of alveolar type 2 pneumocytes can impair surfactant production, causing decreased surface tension and alveolar collapse, as discussed above. Disruption of the alveolar wall barrier can also allow fluid to enter the alveolar space, additionally decreasing the gas exchange surface area. For the remainder of this talk, we will focus on the first 3 levels of vascular impact during SARS-CoV-2 infection. The direct impact of SARS-CoV-2 to the endothelium occurs when the virus directly infects endothelial cells. Endothelial cells in the alveolar tissue, and more specifically the capillary endothelium, do express the ACE2 receptor, as shown by immunohistochemistry, suggesting that these cells are permissive and susceptible to SARS-CoV-2 infection. Additionally, ACE2 is expressed on endothelium of the brain and in vitro by HUVEC cells, the primary human umbilical vein endothelial cell line. mRNA for TMPRSS2, the surface protease that facilitates SARS-CoV-2 entry into host cells, is also detected in HUVEC cells and endothelial cells of the respiratory and digestive tract. SARS-CoV-2 in vivo infection of endothelial cells has been demonstrated by immunohistochemical staining for SARS-CoV-2 spike protein, revealing granular cytoplasmic distribution in endothelial cells derived from patient tissue sections. Transmission electron micrographs of skin and lung sections from COVID-19 patients also display virus-like particles inside endothelial cells in both tissue types. The indirect vascular impact of the SARS-CoV-2 infection begins with the innate immune response. The innate immune response in the alveolar space is initiated by the intracellular cytoplasmic sensors, signaling the detection of pathogen-associated molecular patterns associated with viral infection and replication. This initial response produces chemokine signals from the resident alveolar cells that recruit innate immune cells, such as neutrophils and inflammatory macrophages, to the site of infection. As these cells move along chemokine gradients towards the endothelium, they'll upregulate factors involved in rolling and adherence, slowing their movement as they approach the site of infection. At the site, immune cells will transmigrate through the endothelial barrier and into the infected tissue. When there is an abundance of immune cells transmigrating into the site, the barrier function of the endothelial cells can be compromised or damaged, leading to vascular leakage into the interstitial space or even into the affected tissue site. If the alveolar barrier function also becomes compromised, this leakage can cause pulmonary edema to occur where fluid fills a portion of the alveolar airspace. The vascular leakage results in an upregulation of the coagulation cascade and recruitment of platelets to restore the barrier. At the same time, the loss of alveolar pneumocytes caused by either direct infection from the virus or off-target damage from recruited immune cells can lead to a loss in surfactant production and decreased surface tension, leading to alveolar collapse. Once this occurs, there's a partial loss of gas exchange surface area, and unchecked will eventually lead to tissue ischemia or overall hypoxia as increasing portions of the lung are damaged or become collapsed. This immunopathology cascade results from a dysregulated and abundant immune response to the viral infection. When this occurs, the patient experiences what is called acute respiratory distress syndrome, a morbidity associated with moderate to severe COVID-19. During this process, the virus can easily move from the site of infection, the alveolar space, into the vasculature, where it can infect ACE2 expressing endothelial cells and move into systemic circulation, spreading the infection to other organ systems. With vascular leakage activating the coagulation cascade, microthrombi are deposited into the vasculature to restore homeostasis. The dysregulated clotting and increased recruitment of inflammatory cells leads to blockage of small blood vessels in the tissue. This blockage prevents oxygenated blood from reaching those tissue sites, leading to ischemia and increased cell and tissue death. To compensate for this decreased oxygenation and prevent further ischemia from occurring, angiogenesis mechanisms are upregulated, producing new blood vessels. Increased angiogenesis is typically associated with sprouting angiogenesis that occurs during several well-characterized stages, including upregulation of angiogenesis factors and protease-mediated escape of endothelial cells from existing vessels, allowing the formation of new blood vessels from the existing vessels. However, in COVID-19 patients, there is a noted increase in intussusceptive angiogenesis in the lungs. In contrast to sprouting angiogenesis, intussusceptive angiogenesis is a rapid adaptation of the existing vascular network that does not require immediate endothelial cell proliferation, but instead emerges due to a reorganization of existing endothelial cells and the invasion of endothelial precursor cells. Intussusceptive angiogenesis in COVID-19 lungs is associated with a deformation of the vessel architecture and a loss of the typical vessel hierarchy. Overall, these initial direct and indirect impacts of SARS-CoV-2 infection to the endothelium suggest that we need to further our understanding of the vascular impact of COVID-19.
Thank you, Kristen, for the detailed introduction and for really nicely setting us up for the rest of the presentation. So in this section, I'd like to review the gap in our current resources or in vitro vasculature models. So to better understand direct and indirect impacts of SARS-CoV-2 on vasculature, it's really important to have a physiologically relevant in vitro model, which can be applied across multiple fields, from basic and applied research to therapeutic and vaccine development. While lately there's been a movement towards physiologically relevant 3D cell culture, the majority of in vitro vasculature models are traditionally 2D static cultures. Now, these cultures have many drawbacks, including the lack of perfusion, which we know is important for maintaining endothelial cell morphology and barrier function. Another is the presence of artificial membranes, which act to support the separation of an endothelial cell layer from, say, an apical and basal compartment. And these membranes typically have small pores, which can interfere with the sensitivity of assays as well as compound transfer. Further, some commonly used platforms also suffer from low quality of imaging which makes the evaluation of cell morphology and staining challenging. Other drawbacks of current models include being low throughput, having increased reagent consumption due to the amount of media and/or cells that are required to prepare the culture, And finally, for many of the more complex in vitro systems, compatibility is often a challenge, especially those microfluidic systems, which tend to require special lab equipment, including pumps or tubing, that really makes the adoption of that new technology unattractive. And so MIMETAS has developed a platform that, when paired with the BioTek automated imaging analysis, will help to sort of bridge the gap in our current resources and enable the use of a high throughput, physiologically relevant in vitro vascular model. And that platform is the MIMETAS organoplate. So it's similar to other 384 well plates, microtiter plates that you may have worked with before. However, the bottom of the plate is 2 layers of microscope quality glass, which contains microfluidic channels. And using a pipette, you can actually add cells, extracellular matrix gel, media, and sort of build your stratified tissues layer by layer in the microfluidic channels. And within 1 2-lane organoplate, you can create up to 96 individual tissue chips. So let's go ahead and zoom in on one of those chips. A single chip is made up of 4 wells in a row that are connected by microfluidic channels. The first well is an inlet where you can add your extracellular matrix gel, which fills the channel. The second and fourth wells are the perfusion inlets and outlets where media with cells can be added to fill that perfusion channel. And the third well is the observation window where we can use microscopy and imaging techniques to capture each of the miniature tissues. So it's important to point out that there's no physical barrier that separates the perfusion channel from the gel or the ECM channel. Instead, what runs between the channels is a short ridge of plastic called a phase guide. And so this phase guide actually acts as a capillary pressure barrier. And so you can sort of pipette in your liquid ECM gel, and the capillary pressure barrier will pin the liquid within the channel, and it won't be able to spill into that adjacent perfusion channel. So once the gel solidifies, we can actually add our cells in media to the perfusion channel, and then those cells will be in direct contact with the gel. So in the next few slides, I'm going to share with you some screenshots from a video that will nicely bring together everything I just described and give you a better idea of how a scientist might use the organoplate. So here's a view from the top of the plate where we have a scientist using a repeat pipetter to fill the individual gel channels with extracellular matrix gel and cells. Here's a view from the bottom of the plate where you can see the scientist is working down the column to fill the gel channels with the repeat pipetter. As the capillary forces carry the gel through the channel, here we see a cross-section of the channels, and you can see that the phase guide in between the two is preventing that gel and cells, the red gel and cells, from entering the perfusion channel. You can kind of see that pinning effect. In this case, there's cells embedded in the gel, and they maintain an equal Z distribution as the gel solidifies. And once the gel solidifies, we can add media to the perfusion channel using a multi-channel repeat pipetter. Capillary forces pull the media into the channel. And here we have a cross-section view. The medium is then in direct contact with the gel and the cells. And here's a view from the bottom of the plate with both channels filled. And so during perfusion, the nutrients within the media are able to diffuse into the gel channel and allow for nutrient and waste exchange between the cells present there. We can achieve perfusion in the organoplate with a programmable rocker called the organoflow. The perfusion inlet and outlet wells act as media reservoirs, and you can take the plate, place it on the rocker in your incubator, and the rocking motion drives the fluid between the media reservoirs through your microfluidic channel, and this thereby introduces fluid flow into your culture. The angle and the time intervals of rocking can be modified to change the flow rate to best mimic the flow rate of your tissue of interest. And so in addition to embedding cells in the gel, we can also grow cells against the gel, which oftentimes when cells are seated there in the perfusion channel against the gel, they will form a tubule-like structure mimicking a 3D structure like a blood vessel. And the next few slides are screenshots from a video showing how to build a tubule structure in the organoplate. So here we're viewing the bottom of the plate, and we typically start by adding gel to the gel channel and allowing it to solidify there. Following which we can add cells in medium to the perfusion channel. Here we have a view from the bottom. Now we're back to a cross-section view where you can see the cells in the medium on the left, in the blue medium, in that perfusion channel against our solidified red gel. Now we're back to the bottom view, and we can see the red ECM on the top and the cells in the blue medium on the bottom. And after you add your cells in the medium, this is when you can actually flip and invert your plate to allow the cells to attach to that ECM. And once attached, we can begin perfusion, and the cells will begin to grow and form a tubule structure within that perfusion channel. And here we have a 3D reconstruction of a kidney tubule, which has been stained and imaged. And in the cross-section, you can actually see that there's a lumen formed within that tubule structure, within the perfusion channel, with the phase guide between the tubule and the ECM gel. So there's a variety of endothelial cell models that can be built in the organoplate, and the simplest model is growing an endothelial cell tubule against our ECM gel in the 2-lane organoplate where you can have up to 96 replicates. If you're interested in a more complex model, the 3-lane organoplate is available where we have 9 wells that connect in a square to make a single tissue chip. And so this totals 40 chips on a single 384 well plate. And in that 3-lane organoplate, not only can you culture an endothelial cell tubule against the ECM to mimic a blood vessel, but we can also model angiogenesis by inducing sprout formation from that parent blood vessel. In this case, you would first add endothelial cell tubule to the top perfusion channel, following which you can add angiogenic cocktail of small molecules in medium to the bottom perfusion channel. And a gradient of factors is then formed in the culture, and this allows for the formation of angiogenic sprouts from that parent vessel through the ECM. Another organoplate type which enables 3D vascular cultures is the organoplate graft, with 6 wells connecting to make a single tissue chip and a total of 64 chips per plate. Similar to the 3-lane organoplate, you can add an ECM gel to that middle channel and endothelial cells to the side perfusion channels to form blood vessels. However, the graft is a bit unique in that the middle ECM channel is actually open on the top, so that it allows you to place a piece of tissue, say a spheroid or an organoid, and ultimately be able to vascularize that tissue. So here we have a cartoon of how to build organoplate graft cultures with the top-down and cross-section views provided. So we start off by adding your endothelial cells against the ECM and allow the cultures to perfuse for a few days and to form the blood vessels in those perfusion channels. Then we can add the angiogenic sprouting medium to that middle graft chamber on top of the ECM and induce angiogenesis. And once that vascular bed is formed, we're able to place a piece of tissue or spheroid on top of that vascular bed, and ultimately, with more time, that piece of tissue itself becomes vascularized.
As Kristen just spoke about, the MIMETAS products offer the ability for quick and easy analysis of your vasculature formation via microscopy and imaging. BioTek automated microscopy instrumentation offers modular and upgradable systems designed to suit the needs of the user and provide automated imaging and analysis. There are two lines of automated imaging instruments: the Citation Cell Imaging Multimode Readers and the Lionheart Automated Microscopes. While the lines offer different options and configurations, all BioTek imaging instruments are true microscopes, providing a variety of imaging applications and compatibility with a range of supported labware, including slides, dishes, chamber slides, cell culture flasks, and low-density and high-density microplate formats. Additionally, all instruments provide onboard temperature control, 4-channel imaging with bright field and fluorescent imaging standard across all instruments. Configuration-dependent options include color bright field, phase contrast, and high contrast bright field imaging modes. Additionally, BioTek offers a range of imaging accessories, providing the user with the ideal configuration for their applications. These include over 22 fluorescent filter cubes and color-matched LEDs, ranging from UV to near infrared, and Olympus and Zeiss objectives. The Citation Cell Imaging Multimode Readers offers a unique design, providing configurations that allow automated microscopy and conventional microplate detection in one modular and upgradable platform. For imaging, the Citation systems offer onboard capacity for up to 6 objectives with compatible magnification from 1.25x to 60x air. For workflow automation, the Citation instruments are compatible with BioTek automation accessories, along with additional third-party automation systems. For live cell workflows, the gas control module can provide both carbon dioxide and oxygen control. And the optional dual reagent injector module provides automated injection of up to 2 compounds, integrating injection with imaging or detection analysis. In contrast, the Lionheart automated microscopes provide a compact and ocular-free design and offer a wide range of imaging capabilities in a modular and upgradable platform. The Lionheart systems provide onboard capacity for 6 objectives, with compatible objectives ranging from 1.25x up to 100x oil. The Lionheart FX is ideal for live cell imaging with optional CO2 and O2 control and humidity chambers available. For rapid kinetic assays, the Lionheart FX is compatible with the dual reagent injector module. BioTek automated microscopes produce high-quality images full of rich phenotypic data to move your applications forward. Image capture options include endpoint or kinetic capture, capturing a single image, choosing capture beacons, or montaging across a large area, capturing a single focal plane, or imaging 3D biology with Z-stack imaging. Depending upon the instrument configuration, images can be captured in fluorescence, bright field, color bright field, phase contrast, or high contrast bright field. Together with BioTek's automated imaging instrumentation and hardware, the Gen 5 imaging and analysis software creates the unique augmented microscopy experience. Augmented microscopy is the integration and automation of all steps from image capture to processing and analysis in the Gen 5 software, eliminating the need for post-processing, post-analysis, or additional software, and providing publication-ready data. With augmented microscopy, the imaging workflow is fully automated to increase throughput while providing both qualitative and quantitative data. In this example workflow, the researcher has transfected cultured fibroblasts with a vector expressing GFP. The end goal of this assay is to use imaging to determine transfection efficiency. During the capture step, they are capturing a single image in the center of the well, as described in their protocol. To optimize their image for downstream analysis, in processing, they add a background flattening step. To analyze their images for transfection efficiency, the researcher uses the Gen 5 dual masking technique and applies a primary mask onto the DAPI signal. This primary mask will provide a total cell count based on the DAPI signal, but also identify each nuclear signal as an individual object or event. From there, they are applying a secondary mask to the cytoplasmic compartment, allowing the researcher to determine the total GFP signal within the cell. Once the primary and secondary masks have been defined, the researcher can decide how they want to define their subpopulation of GFP-positive transfected fibroblasts based on a variety of object-level metrics and control images. Here, they have defined their cells as GFP-positive using a mean GFP intensity threshold in the total cytoplasmic compartment. Once defined, cells in the subpopulation can be visualized on an image level and by scatter plots and histograms. To normalize their data, the percentage of GFP-positive cells can be calculated by adding the ratio calculation in our post-analysis options. This augmented microscopy protocol is applied to the full plate of cells, providing data output for any chosen populations and metrics that can be easily exported into Excel. While this protocol is the best choice for their assay, there are a variety of capabilities in the Gen 5 software, as we have discussed. These include the additional capture modes and additional capture methods, additional processing methods that include deconvolution, digital phase contrast, image stitching, or Z projections for 3D biology, and kinetic frame alignment for kinetic captures. The Gen 5 dual masking technique allows for many options in image analysis, allowing distinct analysis of signal between nuclear and cytoplasmic compartments, thresholding, custom object-level metrics, and displaying data in both histograms and scatter plots. Additional data reduction tools for post-analysis methods include ratios, deltas, normalization, custom equations, and methods for qualitative analysis and quality control.
Now we're going to jump into how to use imaging to monitor the growth and morphology of your tubular and vascular bed endothelial cultures in the MIMETAS organoplate. Here, we used a 2-lane organoplate to grow immortalized endothelial cells called HMEC1s against a gel. And over time, you can track the culture morphology easily using phase contrast imaging. And we captured these images using the BioTek Citation 5. You can see on day 0, when the cells are added to the perfusion channel, that they have not yet adhered to the ECM or to the channel. And so after the cells have finally attached, over time, you see a nice progression from days 1 through day 8 of the endothelial cells forming a healthy confluent monolayer. You can also use bright field imaging with the Citation 5 to monitor the culture morphology. And here we have a vascular bed of HUVEX in the organoplate graft, and we can see the progression and growth of the vascular sprouts towards the middle graft chamber over time.
Using the MIMETAS products, the organoplate and organoplate graft, to produce endothelial cell tubes and vascular beds, we can better understand the interaction of SARS-CoV-2 and the endothelium in a biologically relevant model. In the following sections, we will walk through a variety of virology-specific applications and examples made possible by the combination of MIMETAS technology and BioTek automated microscopy and instrumentation. Using these models, let's first investigate the direct impact of SARS-CoV-2 on the endothelium. Prior to infecting cells, it's important to determine if a host cell type is permissive and susceptible to viral infection. For any viral infection, permissivity and susceptibility will require the presence of the receptor, allowing the virus to enter the cell. Using qPCR, we confirm the presence of both ACE2 and TMPRSS2 transcript in the endothelial cell tubes grown in the 2D organoplate and the vascular bed growth in the organoplate graft, suggesting these cells will be permissive to SARS-CoV-2 infection. To confirm the susceptibility of endothelial tubes to the virus and determine viral infectivity, endothelial cell tube cultures were infected with a fluorescent reporter strain of the virus and incubated to allow infection to progress. Prior to imaging, a live cell nuclear dye was added to allow quantification of total endothelial cells and normalization of infection. Using the BioTek Citation 5, we developed an imaging protocol to capture the full endothelial tube in situ as a montage Z-stack. With augmented microscopy, captured images were processed and analyzed for the total cell count, GFP-positive cell count, and the percentage of the GFP-positive subpopulation. In the absence of immunostaining reagents or a reporter strain of the virus, many viral infections can be detected by observing a notable phenotypic change to infected cells, termed the cytopathic effect, or CPE. CPE is typically seen as either cytotoxicity, the rounding of infected cells and subsequent loss of attachment, or syncytia formation, where the infected cells fuse together. CPE can be used as an indicator for infection in the absence of virus-specific reagents or as a screening tool. The presence or absence of CPE is typically indicated as a binary plus or minus score when screening wells. In this example, we analyze non-infected and infected endothelial cell tubes for the presence of CPE. For this virus, CPE is indicated by cytotoxicity and a loss of cellular attachment and confluency. To provide automated analysis for CPE, we determine the sum area of total endothelial cells present in non-infected and infected tubes, and this sum area was normalized to the sum area of the non-infected tubes, providing a normalized percent confluency output. Based on a confluency threshold that we determined, we converted this percent confluency to a binary CPE score, where confluency less than 75% indicated the presence of CPE and thus the presence of virus, and was scored a 1, whereas non-infected tubes were scored a 0.
So using the organoplate 3-lane, we can also evaluate barrier function. And in this case, you can grow your endothelial vessel in the top perfusion channel, and once that barrier has formed, you can add a fluorescent dye, following which, if all of the tight junctions are functioning and intact, then that dye will remain in that top perfusion channel and will not appear in the gel channel. However, should the tubule have a compromised barrier function, say due to cytokines or a toxic compound, then you would expect that the fluorescence would leak into that gel compartment. And here we have an example below using 155 kilodaltons Fitzy dextran, which were captured using the BioTek citation 1. We always use a no-cell control to understand how the fluorescent dye moves through the ECM without any cells present. And that's exactly what we see here. Next to that is an example of a leak-tight tubule where our endothelial cells are forming a complete barrier and preventing the dye from entering the gel channel. And finally, there's an example of a leaking tube where you can see some of that fluorescence is making its way into the gel channel. And we can measure this over time, and typically we can graph these data by dividing the fluorescence intensity inside the gel compartment by the fluorescence intensity inside the perfusion channel. And a leak-tight tubule will maintain low values over time, while a leaky tubule will sort of increase over time towards a value of 1. So in addition to measuring barrier function, a direct result of endothelial cell tight junction function, we can also evaluate the expression of endothelial markers by immunofluorescence. So we can fix and stain your cultures directly in the organoplate. And here we can see robust V cadherin in green and von Willebrand factor in red in our HUVEC tubule.
Now that we have discussed the basic applications to study the direct impact of the virus within the endothelium, let's move on to the indirect vascular effects of SARS-CoV-2 infection. In this section, we'll review how immune cells interact with the endothelium and some sample applications to study this interaction. As we discussed earlier in this talk, immune cells are recruited to the sites of infection and damage via cytokine signaling and chemokine gradients. For this example, we will focus on the recruitment of neutrophils, the body's first responders. In this illustration, the virus has infected the alveolar macrophage, the resident macrophage of the lung. The presence of virus in the cytoplasm of the cell and the replication byproducts trigger cytoplasmic sensing mechanisms, leading to the production of pro-inflammatory cytokines and chemokines. There is a gradient of chemokine concentration with the highest concentrations proximal to the infected cell and decreasing with distance. The circulating neutrophils follow the chemokine gradient from the systemic circulation and toward the alveolar capillary network. As the chemokine concentration increases, the neutrophils upregulate their cellular adhesion molecules and move from the free-flowing lumen towards the periphery. At the endothelium's surface, the neutrophils begin to roll along the chemokine gradient and then adhere to the endothelial cells. They will then transmigrate through the endothelium and through the alveolar epithelium as well, accessing the alveolar space. Once they have accessed the site of infection, the neutrophils can release their inflammatory byproducts and set to work eradicating the virus. Under typical conditions, the virus-infected cell is destroyed and additional immune cells are recruited to resolve inflammation and damage. The MIMETAS organoplate can be used to assay for immune cell adherence to endothelial cells and better understand the role of various inflammatory signals in this process. MIMETAS has confirmed the ability to model this immune cell adherence in the organoplate, as here they group human coronary artery endothelial cells in the perfusion channel. Once confluent, they treated the cultures with increasing concentrations of TNF-alpha for 4 or 16 hours. After TNF-alpha incubation, calcine AM stained monocytes, shown in green, were added to the endothelial tube, with nuclei shown in blue, for 15 minutes, following which they were washed and remaining green monocytes were considered to be adhered to the endothelium. TNF-alpha promoted the adherence of the monocytes at 4 and 16 hours at various concentrations. As many inflammatory cell byproducts can cause off-target damage to surrounding cells and tissues, it's necessary to be able to assay for cell death in this model. Here, endothelial cell tubes, either non-infected, infected, or control-treated, were stained with Hoesch and propidium iodide. The tubes were automatically imaged using our Gen 5 imaging protocol as previously described, followed by cellular analysis. Cell death was determined by propidium iodide staining and quantified as the total propidium iodide positive cells, the percent of propidium iodide positive cells, and the percent of endothelial cell tube confluency. As shown, the infected cells exhibited propidium iodide staining and increased cell death, while tubes treated with the positive control exhibited an even higher level of PI staining, along with a more severe loss of confluency.
Now we'll move on to how the MIMETAS and BioTek platforms can be used to investigate vascular impact level 3 by modeling changes in angiogenesis. So here we've added HUVEX to that top perfusion channel, and once the tubule has formed, angiogenic factors were added to the bottom perfusion channels to promote sprout formation and growth through that gel channel. Cultures were treated with increasing concentration of that angiogenic cocktail sprouting mix, and you can see a dose-dependent formation of the sprouts in the ECM. And we captured these images using the BioTek Citation 5, and what's really great about the instrument and the Gen 5 software is our ability to quantify those vascular sprouts. So using a cell mask, Gen 5 is able to calculate the area and the confluency of the sprouts, which is fantastic. While we have not yet evaluated the contribution of SARS-CoV-2 or hypoxia to angiogenesis in the organoplate, this assay and data analysis package would enable such studies. In addition to altering the concentration of angiogenic cocktail, we can also observe a time-dependent formation of those angiogenic sprouts in the organoplate graft. And here you can see the vascular bed is forming over time, and a tissue explant is added, following which that tissue becomes vascularized.
In conclusion, SARS-CoV-2, the causative agent of COVID-19, has resulted in an ongoing pandemic and global health crisis. While we are still learning about the pathogenic mechanisms of the virus and the resulting disease, multiple characteristics of SARS-CoV-2 have contributed to this rapid spread and morbidity and mortality associated with the disease. Though COVID-19 has been considered a respiratory disease, in moderate to severe patients, multiple organ systems are affected, indicative of systemic spread. Systemic infection is associated with severe sequelae and increased morbidity and mortality. Current research suggests that COVID-19 may actually be a vascular disease, where viral infection of pulmonary endothelial cells can lead to a direct and indirect effect of the virus on the pulmonary and systemic circulation. This new direction allows researchers to expand their focus outside of the respiratory system and explore additional preventatives and therapeutics aimed at preventing vascular infection and subsequent damage and dysregulation. The goal of this presentation was to provide researchers with a toolbox to better understand this virus-host interaction in a high throughput and automated fashion.
From the technology side for MIMETAS, I'd like to conclude with just a few take-home messages. The first, that the organoplate vasculature models address many of the drawbacks of other in vitro models. Next is that there are many assays available to evaluate endothelial cell phenotype and function. And finally, I think we showed nicely in this presentation that the organoplate is quite compatible with the BioTek automated imaging platforms.
BioTek automated imaging and microscopy instruments are available in a variety of modular and upgradable platforms to meet the needs of your specific research. With Gen 5 imaging software and the process of augmented microscopy, imaging protocols can be fully automated for capturing, processing, and analyzing your data, efficiently increasing throughput and efficacy. Together with these MIMETAS products and organoplate formats, we hope to offer researchers a complete toolbox to better understand the vascular impact of SARS-CoV-2 infection.
As always, MIMETAS and BioTek instruments are here to support our customers during this time and offer solutions to help in your research efforts and to accelerate your work. We thank you all for your attention, and we're happy to answer any questions you may have.
Thank you, Drs. Bircsak and Olson, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Let's get started. Our first question is, for cytopathic effect, you looked at a loss of confluency as an indicator of CPE. Can you also quantify cell rounding on a cell-by-cell basis?
Yeah, so that's a really great question. In addition to being able to look at that CPE indicator by a loss of confluency, we can apply a variety of metrics to detect cytopathic effect in your infected cells, depending on what that phenotype is. So that includes things like cell rounding or syncytia formation, or even darkening of the cells as they start to lose attachment. That allows a nice focal detection of viral infection early on also.
Okay, awesome, thank you. Our next question, did you evaluate automated liquid handler to pipette in organoplate, and especially the Agilent Bravo?
That's a great question, and I can field this one, Kristen. So here at MIMETAS, we've actually worked together with an academic partner to use the Agilent Bravo and successfully loaded cells both in a gel as well as against the gel, loading nearly, I would say, almost 100% success rate. And so in addition to loading the cultures, we can actually use the Agilent Bravo to change the culture media, add assay reagents, and things like that. So it's a really nice option to kind of pair all three of the platforms even.
Okay, thank you. We've got another question here from the audience. They say it's a really interesting presentation. And have you used BioTek imaging for herpes virus research?
Yeah, so I'm not sure if we've directly used it for herpes virus research, but we have worked with researchers with a variety of viral infections. And I'd be more than happy to discuss this kind of one-on-one during the networking lounge and go over some of the assays and applications that you might want to perform with the BioTek imaging.
And another question here, acute kidney injury is a morbidity associated with COVID. Could we use these MIMETAS vessels to also grow kidney tubules?
In fact, you can. So similar to the way you would have seeded endothelial tubules, you can sort of seed your proximal tubule cells into that perfusion channel, and after they undergo perfusion, they'll form a 3D proximal tubule-like structure within the perfusion channel. So definitely feasible.
Okay. Another question from the audience. What percentage of non-infected cells end forming leaky tubules?
So that's a good question. So cells that are non-infected or not impacted by toxic insult should form completely leak-tight barriers. So that's 100% of the cells that are not infected form leak-tight barriers. And it's not until we see that insult do we see any leaking.
Okay, got a couple more questions. Kristen mentioned that she used both Citation 1 and the Citation 5. What is the difference between the two instruments, and is one better than the other for organoplate imaging?
I'll field that one. So the differences between the Citation 1 and the Citation 5. So both will work for your imaging and the organoplate. They're just slightly different configurations. So the Citation 1 is an affordable option for automated imaging and also multi-mode microplate detection. There's a limitation in the number of objectives that it can hold compared to the Citation 5. So it can hold 2 objectives. The Citation 5 can hold 6. And there's some additional limitations in the types of imaging modes. So with the Citation 5, you have the full range of configuration options. And the best way to figure out which one of those instruments would be great for you is to just discuss it with a biotech sales representative and a field application scientist, and we will help guide you to the right configuration based on your assays and applications.
Okay, great, thank you so much. We've got another question. In addition to evaluating barrier integrity, is it feasible to measure TEER, transendothelial electrical resistance, within the organoplate platform?
That's a good question. Yes, so we've recently developed a TEER device. You can actually place these, it's sort of like a plate lid that goes on top of the lid, and there's tiny electrodes that go into each well. And all at the same time, you can measure the TEER value for each of your cultures. And so this is extremely compatible right now with our epithelial cultures. And we're working towards making it compatible with our endothelial cultures and hope to have that done by the end of the year.
All right, we've got one more question. We are hoping to analyze the impact of infection on endothelial cell tight junctions. How would you analyze the loss of tight junctions with microscopy?
Yeah, so that's a great question. And Kristen actually showed some data for this. But one of the best ways to do that with endothelial cells is to stain for VE cadherin, and then along with your nuclei and any other cell markers that you'd like. And then we can look at the total VE cadherin staining through a variety of different metrics. So we can look at the confluency of that staining, the intensity, and also look at different ways of analyzing that as it breaks down. So yes, definitely something you can do. And immunostaining is going to be the way to go.
Well, thank you, Dr. Kristin Bircsak and Dr. Kristen Olson, for your time today and for your important research. Before we go, I'd like to also thank the audience for joining us today and for their interesting questions. Questions submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand through July of 2021. Until next time, goodbye.
Bye.